honoured to be asked to take part in this conference and to be invited back into the organic phosphorus fold, so to speak, albeit temporarily. Thanks everyone for the warm welcome. My last publication on the topic of phosphorus was published almost 25 years ago. I was somewhat humbled by this invitation, as my knowledge of current research output is rather scant. So it has been with very great interest for me to learn about your recent research. I've been asked a number of times, how did I get into soil phosphorus research? Why did I work in woodlands? Well, after I'd finished my PhD, I managed to get a job in 1968 in the woodland habitat team of the Nature Conservancy. This was an earlier version of the current Centre of Ecology and Hydrology, and I was at the research station in Grange-over-Sands, southern Lake District. My research brief was quite simply to work on soil organic matter. Soon after starting, I found myself involved in a collaborative whole ecosystem study of a local woodland called Meathop Wood as part of the International Biological Programme. I became involved with a group of specialist researchers from the research station and from various universities in investigating the woodland biomass, tree and ground flora productivity, organic matter decomposition, soil microbiology, soil fauna and nutrient cycling in great detail. It was during the work on these nutrient aspects that I realised that phosphorus was the key limiting nutrient in many ecosystems. For example, forest, heather moors and upland grasslands here in the northern UK. I soon found myself constructing, out of project data provided by other researchers, a P cycling diagram for the whole woodland, giving amounts of P in kilograms per hectare in component biomasses, including estimates of total organic and available P in the soil profile, and annual transfers of P in terms of kilograms per hectare per year between all the different components, including inputs as atmospheric deposition and losses in drainage. I found this research very fascinating. I even got invited a few years later in 1976 to a high-profile conference held in London entitled Phosphorus in the Environment to present a paper on it, and in fact it was published in 1978. Incidentally, 83 papers have been published on this IBP whole woodland study. The experience gained from the project work gave me a good whole ecosystem perspective for my future soil P work. I also gained a wider perspective from being able to compare the results with other IBP studies, for example, those of Solling and Hubbard Brook projects in Germany and the USA respectively, and with those of other IBP biomes, for example, tundra and grasslands. I think it was all this which switched me on. After this work, I resolved in about 1974 to work on soil organic and available P in other Lake District woodlands, particularly as P deficiency could be having major impacts on woodland ecology. When I discussed this idea with colleagues, they made comments such as, there aren't many good methods to use. There are many awkward soil properties which will interfere with getting decent measurements. And soil variability in natural ecosystem is horrendous, so it will be difficult to get significant results. No significant results, no publications, no future. Challenges indeed. Anyway, despite these comments, I went on to examine the within and between woodland, temporal and depth variation in soils of 4 phosphorus properties in 10 Lake District woodlands. I looked at total, organic, isotopically exchangeable P as a measure of available P, and phosphatase activity. I also measured soil bulk density on all samples taken. Even with a modest level of replication, I was able to detect significant differences between woodlands in all properties. Significant temporal variation in organic P, isotopic exchangeable P and phosphatase activity, and significant depth differences and a number of complex interactions in addition. What is more, the results all seem to be quite sensible and interpretable. If the results were expressed in terms of soil volume rather than soil weight. As a rule of thumb, which came out of the project, was that low-level P availability was associated with low total P per unit volume of soil and a high proportion of the P bound in organic form. Low P availability suggesting plant P deficiency in Lake District woodland soils was later confirmed through the use of the root bioassay technique which I had developed. The bioassay is based on a hunger response of living plant roots. Confirming P deficiency in Lake District woodlands in this way was an important piece of ecological information in itself, but it also provided valuable perspective in relating directly soil chemistry to plant nutrition. Incidentally, the low P per unit volume in these woodland soils, the soils are shallow and stony, as I mentioned earlier, might be linked to the fact that most Lake District woodlands have been coppiced to produce charcoal at roughly 15-year intervals over approximately 3 centuries, possibly resulting in the reduction of P capital in soils. This is a piece of historical perspective. This project helped me to gain good starter perspective in soil P work and gave me confidence to continue with the research theme. It also provided me with a number of satisfying publications in Soil Biochem, Oikos and Journal of Applied Ecology. Bearing in mind my colleagues' comments about the potential difficulties of working in soil phosphorus, my message from this experience is, yes, listen to skeptics, but don't let them win by being negative. I want now to come on to the Soil Organic P review, which was published in 1987, the so-called Blue Book. Why did I do this? My objective was to get a proper overview of soil organic P literature and a global perspective on the subject. The review brought together most, if not all, the references available at that time. By doing the statistical analysis on the data extracted from published literature, I was, I think, able to unlock latent information on organic P in soils, its relation with other soil properties, and how it might vary globally with geology, soil type, land use, and so on. I hoped that the review and the statistical analysis contained within it would provide some useful background perspective on the research topic. I gather from your kind comments that the review has attained these objectives. Just a little historical background here. During the compilation of this review 30 or more years ago, I didn't have the internet to help me access literature. I had to read through Soils and Fertilizers and bibliographies to find references and to get photocopies, over 900 of them, of publications from the British Lending Library. In addition, I didn't have any of the database programs to help me organise the information. I had to use my brain and remember where it was published and when. And I didn't have Excel to organise the data either. I didn't have SAS for statistical analysis on a laptop either. No such thing as a laptop existed at that time. There was no PowerPoint for preparing and printing diagrams either. Some of the diagrams were created using Letraset and graphs were plotted on a flatbed printer. I'm mentioning all these things to re-emphasise that the review was done long ago. So let us fast forward to today. You all now have access to newer, more sophisticated methods to analyse organic P. For example, 31P NMR, HPLC, LC-HRMS. You have many automated instruments for these analyses, making them easier to perform and with potentially better quality control. Computing has developed enormous potential, giving you easy access to database programs, Excel, SAS, or other statistical packages, and to the web of science to get almost instantly the information you need. With this increased technological development and 30 years of more research, you have access to more data and more results. With these much improved facilities, you all should have made lots of real research advantages. So you need now to ask yourselves, how has science progressed over the last 30 years? What have been the key new developments? And above all, where has the science been going, and where does it need to go? Clearly, there is a need for a sequel to the organic peer review I did 30 years ago. Don't look at me. You are the ones with the up-to-date knowledge and awareness of the state of current research. Regarding the latter point, where does it need to go? I feel strongly that there is a need to broaden the perspective of the current research. I feel there is a big missing dimension. Imagine, and I'm sorry, this is quite a clichéd conceptual model. Imagine there is a big box of knowledge labelled Research on Soil Organic Phosphorus. From what I have seen, most, if not all your research topics seem to start off inside this box. Most projects seem to get ever deeper into the box, looking at details of increasing complexity as evidenced by this conference. Other research projects also start off inside the box, but try to look outwards towards trends with external factors, such as across ecosystems, plant successions, or pedogenetic developments. I think at least some of you should start off your projects from outside the box and look in. What do I mean? Some of you should start, for example, by addressing the more important environmental issues of our time. Some possible outline examples are effects of atmospheric deposition, for example. Many UK soils have been subjected to pollutant acid deposition for more than a century. In the last 2 to 3 decades, the amount of deposition has declined steadily. As a result, many acidic soils, for example, the Pennine Hills in northern England, which are mostly peaty top ones, seem to be recovering with respect to pH, some by half to 1 pH unit. What effect has this increasing pH had on P cycling from organic phosphorus? Has it had consequences that have fed through to plant P nutrition in these sheep-grazed upland grasslands, perhaps even changing soil microbial and microfaunal communities? Pesticides. Pesticides, and there are seemingly hundreds of different types, are ever increasingly being used in the environment. These often accumulate significantly in soils and are also dispersed further from the points of release by wind and water into environments beyond the crop. What effects do pesticides, particularly bactericides, fungicides and insecticides, have on P cycling from soil organic matter and organic phosphorus? What do pesticide effects on processes tell us about the role of different organisms involved in these processes? What effects do fungicides have on mycorrhizas, for example? The topic of climate change. Climate change is having major influences on our environment. Not only are soils increasing in temperature, but many are subject to major changes in moisture as well. Wet soils are drying out, or are increasing, and soils are increasingly subject to drought or flooding. There are also indications in the literature that climate change is affecting C to NP ratios in soil organic matter in some environments. Are temperature increases and moisture changes reducing the P limitations to plant growth by speeding up P cycling? Will these effects result in improved vegetation productivity and or changing plant communities and their associated soil microorganisms and fauna populations, particularly in cold climate or even temperate regions. Carbon sequestration in soils. There are some strong suggestions, maybe even government policies derived from it, that there should be some land management changes, possibly in grasslands, to foster the accumulation of carbon as organic matter in soils, as a way of trying to mitigate the effects of fossil fuel-induced rising CO2 levels in the atmosphere. How will the increases in soil organic matter affect pea cycling from the expected organic pea accumulation in the soil and subsequent plant productivity? Biodiversity loss. Biodiversity in the environment is in decline across the globe for various reasons. Many wild community types are how they are because of P deficiency and high organic phosphorus content in soils. 4 examples from England are Lake District Woodlands, upland moorlands on the Pennine Hills, species-rich hill meadows and lowland heathlands. I'm sure there are other wildlife habitats in other parts of the world to which this applies. Plant communities in these places are highly dependent on very specific mycorrhizal associations to enable individual plants both to survive and thrive. We need to know much more about how plants in these communities function with respect to their acquisition of P, very likely to come from soil organic phosphorus via mycorrhizas. Research on pea cycling in these wild plant communities is ripe for scientific development, and information gained could be very helpful in aiding wildlife conservation programs. Excuse me. By citing these examples, I'm not saying you should throw away your P31 NMR machine or your HPLC, but I am saying at least some of you need to stand outside the soil organic phosphorus box and start some new research thinking. You need to broaden the research perspective. It has been suggested at this point I might introduce a few words of wisdom, but I'm not sure that I've got many of those left. Anyway, I can't help thinking that the current research is far too heavily focused on detailed chemical and enzymological studies related to soil organic P and its turnover. Do you really need all that detail? You need to justify the detail before getting it. In my view, research data you are collecting is still quite remote from any direct field application. For wider practical application of the knowledge gained, there will be a need for simple integrating indicator measures of soil P process rates or chemical state. And I see it as your job as scientists to develop these integrating indicators alongside your very detailed research projects. I'm also a bit concerned about the fundamental hypothesis underlying your research, namely that mobilisation of soil organic P can be a long-term sustainable approach to crop production without P fertiliser. This will essentially be soil P mining, and it will lead eventually to reduced overall soil fertility. I think we may need to look backwards for ideas to find out how crop production was maintained in the past. Straightforward approaches adopted then was by rotating soil crops, e.g. the Norfolk rotation in the UK. For tropical reasons, the growing of acacia to increase soil fertility with rotated cropping and repeat cycle of acacia is a well-tried system. We should be investigating soil organic P cycling in these rotation systems. But unfortunately, there's a problem here. The necessary experiments have not yet been established as far as I'm aware. I understand some crop rotation experiments based on the Norfolk rotation are being planned at Rothamsted. Another aspect is soil lining. Soil liming is also a well-known and tested way of mobilising soil organic P in soils. However, soil liming has not been mentioned at all during this conference. The question is, why not? My last comments also relate to the past. Particularly for you younger scientists, you must read and digest, and I repeat, you must read and digest the science of research done years ago, even pre-1980 work, which might not be available via the Web of Science. The science of yesteryear have developed many useful concepts and research results, which you may be in danger of repeating. The work you intend to do may already have been done and published, except perhaps isotope applications. So please do not try to reinvent the wheel. Thank you for listening.